Also, of course, we want to work a bit, even though it's not our main focus to generate protocols how to derive iPS cells, because as I mentioned, there is a lot of work already ongoing by very, very good scientists around the world. But also, we could still optimize some of the protocols to maintain these cells, to make them better and more suitable to our needs using complex cell culture systems. For example, organotypical cell culture systems or different matrices or other changes in the cell culture systems that will help us to have a scalable and then stable and good quality cell culture system. I think these are in a nutshell the main objectives that we would like to address. I think this is a kind of an important slide because of course there are many ways to get there, but I mean I think this is the ultimate question we really would like to have addressed. Why we consider this to be pre-competitive as well? I mean, I think to have more relevant in vitro tools is a no-brainer. That is a very good thing for academic research and also for pharmaceutical industry. It will improve the research tools for the discovery process, of course. It will give us some standardization on the assays. And also acceptable quality assessment for the cells. Oops, sorry. And also, we could exchange technologies and knowledge between the stakeholders within this consortium and also with other consortia. Again, we want to foster those synergies as much as possible and also discuss with scientific external experts and health authorities that will be beneficial for everyone. If an assay is acceptable for also scientific experts outside of the consortium, outside of Europe, and also from the health authorities, that will be, of course, very useful. I think the advances in this basic research area will benefit all of us. I mean, pharmaceutical industry for sure, but I think academic research, SMEs, and also patients and patient organizations. So what do we expect to get out for the research and development process? I think to get a better in vitro system, as I said, is an excellent tool, and that's what we are aiming to do. That we're aiming to do this with several other methods for a long time, but the iPS have opened a new perspective. We can understand better disease biology when we have relevant cells that we could use for this. We can try to elucidate druggable pathways for pharmacological intervention. This still remains within the pre-competitive realm, because how a given pharmaceutical company might then try to address this pathway, this will be after the pre-competitive discovery, but we need to know which targets, which enzymes, which receptors might be involved or might be necessary to be modulated to cure the disease or to improve a phenotype. Also, we can facilitate the evaluation of efficacy, of course, once we know a given pathway, whether we see an effect when it's a treatment. And we can also tailor it to specific populations, patient populations, and not use only cells which are derived from cell lines or from healthy volunteers. Also evaluating the toxicity, the unwanted side effects, also using these cells, of course, that would be a major asset because there will be human relevant data. And with all this, we expect to accelerate the discovery process, which is, of course, very good for the patients because we get to the drugs quicker. It's also good for pharmaceutical industry because we can get to the drugs cheaper or more cheaply. And also would contribute ultimately to the 3R principle of reducing, refining, and replacing animal experimentation. If we have very valid in vitro systems, I don't believe at this point in time we can really avoid completely animal testing, but we can certainly reduce it and refine it by doing appropriate in vitro assays beforehand. So I'm gonna now, I thought how to try to bring over or to convey how we discuss within the pharmaceutical members of the consortium. And I thought, because I'm a pragmatic person really, that it would be good to maybe we discuss a little bit of an architecture of the project. I mean, this architecture is certainly not carved in stone, and it might change, and I hope actually it will change when we get the full when we're writing the full proposal. But however, when we discuss this architecture, we're thinking about the things that we thought that were main tasks and main things that we need to do, and therefore I think it's maybe useful for you to know what would we think are the necessary steps to be performed. So the preliminary structure, we divided the program first in 9 work packages. Again, this is very preliminary, and I very much hope that will change when we get your input. We envisage, and I was discussed this morning, and I very fully agree with that, co-governance of each work package by an EFPIA and academic member, so that one is chair and one is co-chair whenever possible, to optimize the integration of both EFPIA and academic consortia, and also to get the scientific co-ownership of the project. We don't want to have a package, I mean, I wouldn't like to see a work package which is just from academia and one is just for industry, and then the communication might suffer under those conditions. So it would be better if both parts of this marriage, as I was mentioned this morning, are the parents of each work package as well. So the work packages, of course, it is a work package, an important work package considering the size of this consortium, of course, the project management. We were envisaging to have a governance, a chair and co-chair, of course, with the representative, of course, the applicant consortium, an executive committee, which would be the representatives of each entity which is participating, of each participant of the consortium, and a steering committee which would steer the scientific work, and it would be probably by the work package heads. All this, again, is preliminary. It will be then in the consortium agreement, this will be then written on put into a more exact terms, but I think there's a bit of the idea to have this sort of representative. We do believe strongly that we need advisory boards, we need scientific advisory boards. I think that it's a very, very lively field, and I think it's very important to have scientists from outside to give us advice as well. And I think I'll take that advice myself from this morning to try to look into the scientific advisory board as early as possible, so maybe start thinking of some people who might help as soon as we have an applicant consortium. Also, it is important, since we're discussing to use samples from patients, to have ethical committee which makes sure that everything we do is actually compliant to every ethical requirement that we need. And we also have discussed quite at extent to have an IP advisory board. The IP situation for all this IMI, and it was discussed today, and of course I cannot go into details since that's not my expertise, but the IP situation is regulated by the consortium, and what the foreground and background on all this is regulated by the contract. However, in the field of IPS, is a lot of IP which will be outside of this consortium that one might need to take into consideration. If we need to take cells from another group or another consortium, or in different situations, it might be important to keep in mind that there is other IPs outside of each company and outside of our own consortium. And of course, we will progress we will monitor the progress with the regular meetings, of course. So we have a work package too. I put this very shortly. I mean, this is in the proposal. So now it's a quick run on the work package ideas. Sample collection and patient selection is very important. We need to see which patients are suitable, and of course, where will we recruit them, on which cell types we need, and how this will be performed. I think this is a very main part of the project, because without the proper samples to derive the cells, without the proper characterization and the proper patient population, the whole program will be endangered, actually. We need to bank the cells. Again, we need to establish a biobank and also see the viability of a biobank to be then persistent after the 5 years of the project. So I think this will have it will be necessary to discuss quite at length what we need to have there, and which are the requisites, the requirements, which other biobanks are available, and so on. Data management, I think in my experience, is something that goes many times forgotten, but I mean, we are talking about a very big project. We will have a lot of information from patients, patient characterization, we have cell characterization, and we have assay results. And all these needs to be put together so that people can analyze it, can make conclusions based on this data.